We're now going to hear from Andy Sheppard. Andy's currently seconded to DAFF as the co-executive director of the DAFF-CSIRO initiative called Catalysing Australia's Biosecurity. You might have heard of it. He is also the chief research scientist at CSIRO working on biosecurity and invasive species management. We're also joined by Jeff Brosel, the Senior Principal Research Scientist in Biosecurity Research from DAFF, and he's also with the CAB initiative. And Jeff's going to participate in the Q&A session later to broaden our discussion of tech solutions potentially available in the NRM space. But first, Andy's going to talk about the new technologies that can support surveillance and management of NRM pests, weeds and diseases, and specifically 3 new approaches: next-gen eDNA, genetic biocontrol, and spacecows. So over to you, Andy.
Thanks, Justin. Can you hear me all right?
Yes, we can.
Yeah, so I'm going to get, so thanks very much. Just want to acknowledge my co-authors, Andrew Hoskins and Kumran Nagalingham, for providing me with the information for the slide. Really, I'm just going to cover two tech opportunities, one in surveillance for management of vertebrate pests and the other is, which is actually on the ground and running support, and the second one's the genetic control options in not very much detail given the amount of time we've got. Anyway, so in terms of surveillance management for ferals, CSIRO with multiple partners got up a project under the Smart Farming Initiative through DAFF. It's actually a project led by an Indigenous corporation to build a platform to be able to surveil for feral animals for achieving multiple benefits on their land. Lots of partners across Northern Australia, and made quite a bit of media when it pitched itself as spacecows, a landscape scale herd management for unmanaged cattle and buffalo in the Northern Australian Indigenous estates. And as I said, the program is very much run by our Indigenous partners, and CSIRO and other partners like Microsoft provide the technical capacity for that team to build what they're doing. So I'm just going to run very briefly through the project and what it's achieved. It came to an end on the 30th of June, so it's a good time to share what it achieved. So it's really focused on developing a tech-based decision support platform for managing these unmanaged feral herds on country. So it was focused around tech development, both the tech and the data platforms to be able to do the analysis, Indigenous training, particularly around the tech, but also ethical mustering, protection of environmental and cultural values, and to create and support viable on-country business opportunities. But it has a complex context, right? Because these Indigenous corporations are running pastoral and buffalo enterprises. So they're providing into pet meat, the live trade, safari hunting, and crocodile food. So they've got multiple businesses. There's also the importance of these animals for local meat consumption, impact on traditional natural resources like crocs, turtles, and water lilies, impact on environmental values, impact on cultural sites as well, and carbon benefits. So the whole point of the project was to be able to try and capture and support all of these benefits around. So where did we do it? It was done in remote parts of far north Queensland, Cape York, and also on Arnhem Land. So it was innately tough and difficult territory to get out to. Why was it done? Because currently buffalo populations are having a huge impact on northern floodplain wetlands, as you can see from this. So it's around how do we better manage feral animals at a landscape scale to be able to manage for these kind of impacts. And it was built on, as I say, developing a tracking technology platform that would be able to monitor and understand where these feral herds were in the landscape and provide information that would support management decisions. To create effectively the world's largest remote herd monitoring program. And it was built on satellite tracking of using ear tags that were sourced, from off the shelf from the marketplace, but a long process of getting those ear tags out onto the animals and being able to monitor, obviously, a proportion of them in the landscape to be able to understand and predict where the herds were there. So it had quite a complex plan to address all these challenges. So as it was set up, the Smart Farm project was around defining an operating area, making sure that we had a mustering method to be able to be informed by the technology, identifying, develop, and installing the ear tags, having the monitoring and digital platforms to be able to collect and analyze the data, working on targeting particular herds and animals, having to deal with the handling of those animals in order to be able to install the ear tags and releasing them, and also recognizing that there is tag loss and need for tag retrieval. And then also in terms of getting the satellite tagging system to work, it was around making sure the technology worked, activating the tags, putting them in place on the animals, getting those tags to download data effectively through the satellite and on-ground data tracking system, and getting that data back to base and data visualized to provide the information you need. So what kind of movement field data was collected? Well, there were animal density surveys done through helicopters to support the satellite tracking. The satellite tracking itself tracked the movements of the animals across the property in the context of habitat suitability. There was also the tracking of the management activity. So we tracked the vehicles as well that went out and understood the difficulty of terrain they were moving through and how efficiently they could get to particular parts of the property to undertake the management. And from the data was able to forecast herd movements and where herds were in the property for future harvesting decision making through a data analytics platform. The main output from the project is a number of business-specific dashboards that are available and can be used now in real time by the businesses on those properties to be able to manage feral animals. These dashboards provide the forecasts of the herds in real time as to where they are on the property. They also link to meat prices, so the owners can understand where the meat prices are optimal in terms of being able to harvest them to get it into the meat markets. Includes also fuel prices, so you can understand the costs of getting out to these regions to be able to undertake the management. They inform managing efficiency, particularly in the remoteness of these locations, and the viability of harvesting. They also provide you measurement of the carbon benefits based on the number of animals that you've harvested, and measurements also on environmental and cultural benefits that you've achieved through these landscape activities. And now the plan is to scope it out and take the platform to a whole range of vertebrate pests across the north. And here's the first view of how we want to take space cows as a broader feral animal survey to be able to understand and manage where these feral animals are at a much greater scale, and be able to look at Northern Australian scale carbon impacts of harvesting to much better manage our feral animals into the future. Then I'll just second part of my talk's really about some of these genetic control approaches. As I say, I haven't got time to go into great detail, but I'm going to primarily tell you about the two main approaches that are being developed. The first one's called RNA interference, or RNAi, which is a topical way of applying a solution containing double-stranded RNA sequences that switch off genes in your target organism by mediating growth and reproduction. And then secondly, I'll come on to the more widely known and more controversial gene drive systems. So again, I don't have time to go into this in detail, but this gives you basically a schema of how the development of this topical or spray-on RNAi is developed, but it's basically identifying target genes and then developing sequenced RNAi that will can be introduced when they get into a cell, can be introduced and will attach to and switch off key genes. So it's exogenous gene silencing as a way to reduce the fitness and improve the management of some of these pests and diseases. I'm going to talk specifically about some of the work that's actually happening on weeds. I'm actually today at a workshop here in Brisbane with some of our American colleagues around how we can progress RNA management for weeds. But it is also, there's a commercial product available in the US for southern corn rootworm. And some very important work's been done by Nina Mitter at University of Queensland, applying this for the management of viral diseases of plants. So it's a technology that's very much coming onto the market, but from a weeds perspective is still under development. So the Americans have been working on RNAi to control Phragmites australis, or common reed, in the Great Lakes, but through this gene silencing approaches. And they've been looking at genes that to target growth, seed production, vegetative reproduction, photosynthesis, reproductive organ development, and pollen production. But the work we've been doing in Australia has been focused on a much more important weed for us, which is wild radish. And it's one thing to be able to develop your RNAi active ingredient, but you need an effective formulation to be able to deliver that in a sustainable manner into the field where it can persist and continue to be active, which is a lot of work where the UQ group has been doing. But we're at the process where we've just managed to produce proof of concept that we can suppress and knock down growth in wild radish in a laboratory setting. And we're now looking at how we improve the delivery to target cells through the plant epidermis, and also understanding the costs of these kind of herbicides and their scalability. But because this is going commercial, they've now got we know already they've got the commercial viability of RNAi down to about $1 a gram, which is a huge amount of active ingredients. So it's becoming very commercially viable. Second system I just want to talk about briefly is gene drives, which I'm sure many of you have heard about. Even the minister last week, Minister Plibersek, talked about the development of genetic opportunities to control feral cats. So I just want to give you a bit of background. Again, here's the schema for how you build a gene drive based process, which is basically embedding through genetic manipulation genome sequences into your target organism that will reduce the fitness of that organism. So it's a GMO, and having control systems so that the gene drive can perpetuate and suppress populations at a regional or a local or regional scale. The first successful development of a gene drive system was in the malaria mosquito. Again, driving these mosquitoes to a single genotype, which has a much reduced capacity to transmit malaria. And that's a program supported by the Gates Foundation called Target Malaria that's operating in Africa, and the approach is spreading around the world. But where it's been developed primarily in Australia is for control of rodents. This is a diagram of what a myeloid gene drive looks like. You've got your gene drive mouse, and every wild type mouse that any of these gene drive mice mate with, all of the offspring are male. And there are two approaches. One is you can do it all synthetically using CRISPR-Cas9 gene editing, but the approach that's been developed in Australia is actually built on an existing selfish sex gene in mice called T3, where we're trying to augment the presence of that selfish gene in the system. Again, we got through the collaborations, this is work by Paul Thomas at University of Adelaide. We now have proof of concept in the lab for a working gene drive system in a mammal. That's the first time in the world where that's been achieved. So it's a great achievement, and it's an Australian achievement. And Macquarie Uni have also developed a system now that is proof of concept in another vertebrate, which is fish, with the aim of coming up with a solution that may be able to help us long-term in suppressing our feral carp populations. In order to do this kind of work, it's complex, and it requires a lot of interdisciplinary work, a lot of social science. And so we have a big international consortium that's been working together to develop it. We're in the very early stages, and again, the workshop I'm at today is discussing it, of developing gene drives in plants. Plants are much more complicated. They have very complicated reproductive systems, but the thinking and the research is starting. There's actually more work going on in the US than there is in Australia at the moment, but weeds are another option that we're working on. But these kind of technologies are controversial. They're GM, and you need, before you even start, to ensure there's a reasonable amount of social acceptability around the science itself and the risks. And as part of our consortium, and in Australia, we've done a huge amount of work to look at social acceptability of gene drives, particularly in feral cats, and CSIRO's published a report on this, and those and the public acceptability in Australia is pretty high. But you tend to get two responses from the community. One is they think it's amazing and we should do it. We should have done it yesterday, and others that get really concerned about the control of it. So you have these extremes in terms of views that you have to manage. And in order to address that, through the Science Journal, the consortium produced a principles of gene drive research around how we will do it in a transparent and accountable way to ensure that the public understands exactly what we're doing. Both of these technologies have a pathway to deployment here, from conceptualized right the way through to regulatory approval for deployment. This is a very long process, particularly the regulatory approval. Nobody's released a GMO for an environmental benefit. So there's a long process, particularly for gene drives. And these two technologies are different stages through this pathway to deployment. So to summarize, there are plenty of technologies out there that have real potential to help us manage feral pests and weeds. We can't ignore them. We should test them in terms of our context. So our role is understand as science agencies is to understand and regulatory organizations to understand the possibilities, and let society ultimately decide if we want to use it. And that way we can ensure that we can adopt these technologies using a responsible innovation approach. Thank you. Thanks, Justin.
Thanks, Andy. That's fantastic. And it was a bit of speed dating in presentations, but really interesting and exciting stuff.